Hello, DLG4 community. My name is Sabrina Merchant. I'm the Director of Science for the DLG4 Research Fund. It brings me great pleasure to announce that we have awarded a $455,000 research grant to Dr. Alysson Muotri at the University of California in San Diego. Dr. Muotri is a renowned professor of pediatrics and cellular and molecular medicine, the director of the stem cell program, and a member of the Stanford Consortium for Regenerative Medicine. Dr. Muotri has made remarkable contributions to the field of regenerative medicine, including his groundbreaking research in 2020 on brain organoids, which demonstrated how a genetic mutation associated with Pitt-Hopkins syndrome impairs neural development, and his pioneering work using gene therapy to recover the gene's function. His impressive achievements have earned him over $11 million in grants from the California Institute for Regenerative Medicine and prestigious accolades such as the National Institutes of Health Director's New Innovator Award. On a fun note, Dr. Muotri sent thousands of brain organoids to the International Space Station. With that being said, I am delighted to welcome Dr. Muotri to our community. Thank you so much for being here to talk to us today about your research and how it could improve the lives of children and families affected by DLG4 synaptopathy.
Thank you so much, Sabrina. My pleasure. I'm so happy that we are finally working together. I think we have been discussing a lot about what the project would look like. I mean, how to first start understanding the diseases from their neurobiology, their neuropathology, towards like a potential treatment, and everything starts with the first step. And I think this is the first step that we are having now.
Thank you, Dr. Muotri. For background information for our community, could you describe what a viral vector gene therapy is?
Yeah, so once we have a genetic mutation, this is one gene that is mutated in your cells and make the cells malfunctioning. So in the case of DLG4 is a synaptic protein that makes your synapses malfunctioning. So this happens throughout your brain, perhaps more harder consequences in your frontal cortex, which is the core of the computing for your brain, and that creates all kind of sorts of problems. So fixing that would be important because then you can have like a better quality of life. So to do that is a big challenge because your brain is composed by billions of neurons, and each one of them have the genetic mutation in their cells. So how to reach those cells and deliver the correct gene? So we scientists, we learned that viruses can do that really well. And one of the common viruses that we use, it's a flu cold virus type. It's called AAV, adeno-associated virus. And it's a very, if you get that virus, which many people get them, you might have like a very mild flu symptoms. That's it. So it doesn't cause any harm. But nonetheless, what we do is we remove all the genetic material from the virus and we replace with the correct version of the gene. In this case, it's going to be DLG4. And then we use that virus to infect the human body. The virus will go to all the tissues, including the brain, and transfer their genetic material to each one of these cells. Sometimes we don't get more than 10% of the cells, but surprisingly, that seems to be enough to see like an improvement in cognitive function, at least in animal models. And we also see like a good recovery in brain organoid models, which is the human model that is more equivalent of the human brain. So everything suggests that even if we cannot achieve the replacement in all the brain cells with this type of virus, even a fraction of that is already enough to have a benefit to the individual. And of course, I mean, science is never static. We are developing better and better vectors all the time, better tools to infect more cells, to control that delivery in a more precise and systematic fashion. So things are moving along. But the first thing is proof of principle. We first need to show that, yeah, replacing or adding the correct gene inside the cell can actually correct all the downstream genetic alterations caused by that mutation. So that's the first step, and that's where we are.
Thank you. In our community, many parents have asked us about how far along gene therapy really is toward a treatment for our children with genetic, neurodevelopmental, and epileptic disorders. Can you give us examples of similar diseases to DLG4 synaptopathy that have used viral vector technology?
Yeah. So there are a couple of conditions where gene therapy is already a reality, is already happening. And there are many clinical trials where this is also happening. On the neurodevelopmental disorder, there's a couple of them that is on the process. I think the first clinical trial for Rett syndrome is going to start this year. We also hope that our clinical trial for Pitt-Hopkins disease might actually start this year as well. And there are others that are already more along the way. For example, for Angelman syndrome, there are situations where those conditions are moving faster than others. And this is related to the patient advocate, the resources that the investigators have, and how old is the condition? How far do we know about the gene that causes the disease? That's why some of these syndromes have like a fast started. But nonetheless, they are pioneers on teaching us how to do it, and that can be applied to all other neurodevelopmental genetic conditions such as DLG4.
That's very exciting for us. Could you give us a brief summary of the research DLG4 Research Fund is funding with you, and also give us your vision of how this can subsequently progress to a clinical trial for our families?
Yes. So my lab focused on the use of a brain organoid. So to understand how the brain works, I mean, scientists have relied for so long into animal models, such as the mouse. But the mouse brain is very different from the human brain. So there's so much you can learn from the mouse. What my lab is specialized is on the recreation of the human brain using stem cells. So basically, we reprogram cells from patients that carry genetic conditions, and we create brain organoids from them. So in our project here, we're going to have fibroblasts, skin cells from kids with DLG4 mutations, and we're going to reprogram these cells into stem cells. So the skin cells is no longer a skin cell. It becomes a human pluripotent stem cell, meaning that those stem cells can become any cell type in the body. From that stage, we induce the cells to become a brain organoid, which is a three-dimensional structure, tiny, 0.5 centimeter in diameter, that contains all the major cell types of the human brain. And surprisingly, it works like the human brain. It starts to make connections, have like the anatomy that resemble the human brain. So it's a good model to understand what happens in the brains of people with genetic mutations. So because we capture their genetic alterations in those brain organoids, we expect that, for example, the synapses among these neurons would be somehow defective or altered because of that genetic mutation. So we should detect it as a consequence. For example, networks or the communication between the neurons would be affected as well. And we have several tools to measure that. Once we measure that, then we're going to try to rescue or to revert the condition by using these viral vectors to replace the corrected gene back inside the cell. Now, can those neurons, those mutant neurons, now with the correct version of the DLG4 gene, can they start to function like a neurotypical neuron, like a control or a normal neuron without the genetic alteration? So that's what we are looking for. In a positive scenario, and this has, we showed that for many syndromes that this is possible. In a positive scenario, which I'm expecting here, then we have a proof of principle that we can discuss with the FDA to start thinking about a clinical trial, how this clinical trial would be. So most likely, the next following steps after the proof of principle, it is to produce these viral vectors in large quantities so we can infect people, we can infect the human brain with them. And the FDA might ask for some toxicology. For example, they want to make sure that the virus or the new gene is not doing any harm in people. So the first step is really to test that. And we use like a non-human primate, a monkey, and to see if the virus can go to the brain, how much of the neurons is infected, if there is any side effect that we're not anticipating by this virus. So these are primate studies that are highly expensive, We're talking about like $5 million to perform these experiments. But now, at least now here in California, there are agencies that are prepared to support this kind of a stage. Once we clear that, once we know that the viral vector is working and there is no side effect, then we can prepare for a clinical trial. That's the part where we recruit people, and we're going to do like a very small dose of the viruses in the beginning, just to see if everything works, if there is no reaction. And passing that, we go to a phase 2 where we increase the dose, and we're going to see if there is any alteration in behavior or in cognition, any benefit to these patients. So that's already like a phase 2 clinical trial. And then the phase 3 clinical trial is when you test with more people, different mutations, and we include long-term studies just to observe if those patients that receive the gene therapy are showing like signs of improvement and continue to show no side effects. Once that is done, then finally we get the FDA approved to commercialize the gene therapy. And that's when it enters in the market and becomes available to everyone. During this process, we're going to be working with the foundation, with you guys, all the steps to kind of make sure that we are all aligned on the goals and what we think is a acceptable side effect, if any, and if it's acceptable benefit to these patients. So we're going to learn from the natural history of these patients, for example, if the gene therapy is actually working. And then, I mean, at one moment, we will need like a partner, most likely a pharmaceutical partner that has enough funds to kind of push this forwards and have the capacity of commercialization of this gene therapy trial, because this is truly expensive. It's a very hands-on and detailed technology that requires cautions all along the way until it goes to a patient. So we cannot cut corners. We have to do everything very diligent and make sure that there is no harm to these kids in the first place. So that's, in a nutshell, that's the entire process.
Thank you. That was really helpful. And I think I'm really glad you brought up the work of patient advocates and patient families in the natural history studies, because we really want our population to continue to get active in this, to move this forward. Could you also, since you talked about Pitt Hopkins, the viral vector, could you describe the similarities and differences between that research and the DLG4 research grant?
Yeah, with Pitt Hopkins and other syndromes that work in the lab, like Rett syndrome or CDKL5 deficiency disorder, we always start in the same way, which is the basic characterization of the brain cells that has the mutation in those genes. Of course, each gene does a different job inside the cell. So the MCP2, for example, for Rett syndrome is a nuclear protein that controls gene expression. CDKL5 is a kinase that helps to modify other proteins. DLG4 is a structural protein at the synapses. So every gene has a different function in the cells, and that might create different phenotypes in the cell. Phenotypes mean alterations compared to a control or a neurotypical, fully functioning brain cell. So here, for Pitt Hopkins, what we observe is that the mutation causes alteration in cell proliferation and cell migration and the formation of a specific subtype of cortical neurons. And those cortical neurons are reduced in the patients. We even confirmed that using a postmortem brain tissue from one of the cases that unfortunately passed away. But the family was kind enough to donate the brains to us. So we validate that thing. So we see that alteration in there. And now with the gene therapy, what we expect is not only to correct the structural alterations, but also the functional alterations. Because you can imagine, if you don't have all the cell types in your brain doesn't work properly. So the networks are different. So can the gene therapy rescue that? Can we see a rescue of these structural defects and the connectivity between the cells? With DLG4, we expect the same thing. I would even believe that would be a little bit easier to correct the DLG4 than Pitt Hopkins or Rett syndrome, because these are proteins that have act as a downstream pathway affecting many other genes in the network. DLG4, we know more about it. We know it's a synaptic protein. So I think it might be simpler in a way to see a rescue or a phenotype. So we already know what to look for. We're going to be looking for the synapses, and we already know what to expect. So very different from Pitt Hopkins or Rett syndrome or CDKL5, where we have no idea. So we start with a very exploratory side. So here we have the benefit of knowing more what the gene does, so we can focus more, we can target better the experiments.
Thank you. This is really good news. My final question is, what parts of the DLG4 project are you most looking forward to?
Well, I mean, I think the fact that, first of all, there are 3 steps that I think I see as a magical thing, a miracle. The first thing is to reprogram cells from individuals carrying the mutation and making neurons from them. So this is, to me, it's already amazing, because we never had any other moment in history where we could analyze human neurons carrying the genetic material of humans, of patients. So being able to have neurons from DLG4 patients in the lab is going to be a fantastic opportunity to learn more how they behave. Then the second thing is I'm hoping to see, for example, a decrease in the number of synapses. These are the excitatory glutamatergic synapses, where DLG4 might be having like a major function. And finally, what I'm most excited about is to see a potential rescue. Just replacing the gene or putting the correct gene back. Can I correct? Can I make those synapses work again? And can I fix the network? So that would be a fantastic data, and I'll be so happy to have this final data to share with you guys.
We will be too. And thank you once again for your valuable insights and for taking the time to speak with us today. Dr. Muotri, your dedication to advancing research in the field of DLG4 synaptopathy is truly commendable, and we feel honored to have you working with our gene. We can't wait to hear more about your research and see how it progresses in the near future. Thank you.
Thank you. We will be updating you guys as we go. Thank you.